Good afternoon, sir.
Yeah, good afternoon. I'm pleased to have you back here again. Today we'll be looking at topic 2 of ANP504, biotechnology in animal production. Last week we're looking at the basic concepts, but today we'll be looking at the genetic tools that is being used for all of those basic concepts. I will share my screen and take you through the lesson for today. It's all about genetic engineering tools. Like I said, last week we're looking at the GMO food. And so we're looking at all the product that is a function of genetic engineering, recombinant DNA, and biotechnology. And today we look at the tools. And by that tools, we can first ask a question whether it is possible to have identical twin from two mothers without a father. Of course, this is possible. This is what this course is all about. Scientists are able to make genetic copy of a sheep by cloning. They call it dolly sheep. These are examples of a clone of human being, clone of a sheep from the dolly sheep. That is the tools we'll be looking at. A clone is an organism that has the same genetic makeup as another, and it's all achieved by genetic engineering tools. Okay. Now, if you look at, we'll be looking at some learning outcomes to achieve in this lesson. We want to be able to describe, what DNA is and its importance in cloning. We want to look at important steps that we need for marker-assisted selection. I want to be able to describe its usefulness. Now, so when we talk about DNA, we're looking at the basic recipe of our body, okay? And when we talk about assisted age selection, we're looking at steps for making these GMO products. And the question we'll be asking ourselves is, what is their importance? You may say, I don't know anything about this. That's why we are here. So the diagram at the background is what the DNA configuration. What is a DNA? Is a biomolecular, complete information of our organs is contained in the DNA of that individual. Every cell in the living body has a DNA. It is a building block of our body, made up of billions of nucleotides. And how is this DNA used? Well, we manipulate the DNA, to make any product, GMO product we have. Okay. Now, what is a clone? Well, we want to create an individual cell from a progenitor, from their descendants. You're welcome, if you're okay. We're just looking at what a clone is, and yeah, the clone is just a group of cells made from one progenitor. Simple as that. Example is what we showed you before. Woman can be cloned. Sister clone of dolly has been cloned. And that's a dolly sheep. What's the process? This is the process of cloning. If you look at the text, we explain it, in details there. That's the process. Unfertilized egg is where the nucleus has been removed. Egg with no nucleus combined with, a developmental organ taken from the sheep, combined together through an embryo development, a new clone, is developed. A new sheep of the same genetic components is developed. Now, what is the uses of this clone? We use it to make new genes of higher quality, expand DNA sequence, and finally widen the range of biological experiment and practical. When you see the fingerprints, of the genetic importance and protein production, that is the uses of clone. Many of these GMO products are functional. What are the steps? We do fragmentation, litigation, transference, screen selection, and this is the techniques. We have four techniques, used in genetic engineering. Okay, we identify the organism, we extract it, we remove the gene from the rest of their DNA, and we insert into new hosts. Now, what is this tools that we use? We use this marker-assisted selection, and by it now we look at an indirect way of selecting trait that of a very good importance connect with another trait that is that where the productivity is very high, is disease resistant, antibiotic factor. Okay. Now, the commonly used molecular markers are 5 of them. These are the things we use, in details to look at for this DNA, reconfiguration. These are the tools we use for GMO, different type of molecular markers for specific purposes. We'll go into details on other aspects of the topic. These are 5. We have the importance of this selection. We look at the parent, we look at the rate of the breed production, DNA selection, scoring, and correlation of the morphological pictures. And these are the importance of this genetic marker selection. They are very, very important. They are effective in improving the plant and the animal. They are effective in transferring, quality gene from one product to another. They are also very important in transferring male sterility, and of course, the biotic and abiotic resistance future of one animal is transferred to the other. And of course, when you look at this animal, for example, the stress factor is able to resist, accommodate a lot of stress. The embryo quality is high, the temperature body is being able to regulate to have, maximum productivity, and all of that. So these are the things that genetic engineering tools are able to accomplish. In essence, the DNA that contain all genetic information, are able to be used. They are used in configuring all of these things such that we're able to have GMO products that is of benefit to mankind. So these are the things that we are looking at, and I hope that you will take advantage of the lecture notes for the detail of what we want you to know. Okay, so before we let you know, before we let you go, we will let you do a little bit of a test item for you to a lesson, so you can be able to remember everything we have learned. You write down in your own words 5 vital important applications of marker assisted, enumerate their relevance, demonstrate the process of cloning in a well-labeled diagram, and provide a clear definition of different terms used in this subject matter, in this topic. And you can also write down a potential use for the resulting clone animal for your exercise. I hope to see you in the next lesson, where we'll be talking about a different topic entirely. We will brief you. We will send you notification for the next lesson as you take your assignments if you have not done so. So I wish you the best. Thank you. My name will remain Dr. Almate Samuel, your facilitator. Thank you. Do you have any questions? Do you have any question? Okay. I'd like to see your faces. Okay. Your internet is not cooperating. Okay. Okay. Thank you very much for coming to the class. Chris, do you have anything to say with respect to what we have just said? Do you have any question, Chris? Can you hear me? Any contribution? Okay, if you cannot hear me, we'll post this video to you and the lesson notes. Make sure you read it, listen to it all over again. I wish you the best. Bye. Bye now.
Bye-bye, sir. Thank you, sir.